So we have a couple more minutes to people in. So I just wanted to chat. I mean, how's the weather? I mean, over there. I mean, the Texas. I heard. I mean, freezing now, probably back to normal, right?
Yeah, back to normal. Yeah.
And then how about Mattheos? I guess. I mean, freezing today.
It was really bad in January. I mean, it dropped to something like -10 or something. But the past, I would say 1 week, it has warmed up for our standards. It's like around 0 now, like 0 Celsius. I mean, around 35 degrees. So the snow started melting, which is really nice. But we'll see. It is
cold. It's 55 here today in Delaware.
55.
Wow.
Yep. Mm-hmm.
Nice.
It's the same here. We have 3 days snow. I stuck at home for 3 days in a row, as I said earlier. And then now the temperature probably more above 50, and then all snow just gone.
Nice.
I mean, we have the 12-inch snow.
Yeah.
And then I thought 12-inch snow is too much. And then actually I sent email to Crystal Prater saying, Chris, finally I beat you in terms of snow. And then she replied back to me saying, oh, we have the more than 12-inch in the other week. I mean, and then I said, I cannot
right?
be
What
my
do you
advisor.
expect, right?
Yeah, I cannot be my advisor forever. No student cannot, be, advisor forever. That's why,
when I think about it, so when we got a lot of snow as well, but then when you think about it, there's people that live even more north than us. So my brother lives in Ottawa in Canada, right? So they got like double the snow that we got. So there's always someone that's worse than you. So that makes me feel a little bit better.
Okay.
We're not like the worst.
Yeah. Actually, we are very short on food during that snowstorm.
Oh my God.
And we kind of just got everything from the refrigerator, everything, and then on Saturday finally we out, but we kind of on the verge of starving.
Taisok, do we have 1 minute?
Yes.
Okay, I'll be right back. Sorry.
Sure. So while waiting, do you want to test your slide or?
Yeah, sure. I can really quick show it. I mean, if it shows up, it should be fine. Can you see my slide?
Yeah, I see, but can you give us the slideshow?
Yeah.
So slideshow. Okay. So could you go forward? Okay. Good.
Okay. Cool.
Okay.
Good.
Oh, you used the same figure for my introduction.
Yeah. Oh, really? Okay. I just copied from your slides then.
Yeah, it's my best the review paper. In terms of synthetic biology.
Yeah. I really liked it because I'm teaching bioprocessing engineering class. So every time I introduce synthetic biology, I use that toolbox.
Yeah.
Just say, yeah, it's a toolbox that you can use. What can we do with it? Yeah.
You know what? I used that slide for 10 years. I mean, I guess. And then I finally met the author, European person, but I finally met, last year virtually, because she's the European person and I never had a chance. But virtual, format, the beauty of virtual format is basically I could meet somebody I never met before. So.
Oh yeah, that's true. So
okay.
that's a good part.
So everyone ready? We could start.
Yeah, I'm ready.
Okay. So all right. So, good morning, afternoon, evening, hola, hello, bonjour, ni hao, konnichiwa, annyeonghaseyo, and hello all, and welcome and thank you for joining us. Oh my God, the 20th seminar and RPI day. Today is a historic day. In 1947, 75 years ago today, the World War II peace treaties were signed following the end of World War II in 1945. It's called the Paris Peace Treaties. I believe you remember from your history class. And today, many wars or competition are occurring including the Winter Olympics. I also heard multiple unfair umpires manipulate the winning, killing young people's souls. And also, unfortunately, Eric Ronder, the Biden's top science advisor, resigned after acknowledging demeaning behavior. I hope that at least in the game of Olympics, I mean the event of young people's effort and competitions, and in academic research, fair competition and collaboration will be guaranteed. Without fair evaluation, recognition, and sportsmanship, our next generation souls, both Olympic participants and young researchers, would be damaged. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Mattheos Koffas. He is, of course, a professor of Rensselaer Polytechnic Institute. More importantly, he is an editor of many journals, including the editor-in-chief of Metabolic Engineering Communication, one of the best metabolic engineering journals. But even more importantly, he is one of the kindest professor in the world. I'm saying so for sure, because he drank with me during one EIC meeting reception, even when I was just a PhD student, and he was a rising star professor or even established professor at that time, more than a decade ago. So what else do you want to know about him? So Mattheos, the virtual podium is all yours now. Thank you so much for your time today.
Wow, thank you so much, Tessa, for this very generous introduction. And again, I have to say, you have tremendous memory. I wish I was like that. I was actually very flattered when you first contacted me, and you asked me to basically join the seminar series as a pioneer. And this is a wonderful seminar series that you basically put together dedicated to young investigators in the field of synthetic biology. And frankly, I'm still struggling with the concept of being a pioneer. I'm still struggling with the concept of actually being old. So I'm slowly getting there, but so hopefully will happen. So I really wish something like this existed back in 2002, and when I started as an assistant professor, and I think I already mentioned that to you before. So when I agreed to make this presentation as a pioneer, I wasn't quite certain of what I was going to talk about in such a short time, especially since this series has been going on now for a while, and there have been several brilliant people with very thoughtful words of wisdom and lots of experience in the field that have already presented. So what else is there to say, right? So what I decided to talk about, I decided to talk about my mistakes. So the mistakes that I made, especially as a young investigator in the field. And I asked myself last night, looking back to my 20-year-old career, what is the most important, and what I mean by that, what is the most impactful mistake that I made as a young investigator? So in order to answer this question, I think first I need to say why, as a young engineer, I decided to become a synthetic biologist. So I come from a generation where basically molecular biology was not part of a traditional chemical engineering curriculum. So I still remember when I was giving departmental seminars in the mid-90s as a graduate student at MIT, and later when I was interviewing for faculty positions in the late 90s, that I actually had to include slides in my presentation about what is a PCR or what is a restriction digest, because people in engineering departments did not know these things. So I remember also my first metabolic engineering conference. That was the very first metabolic engineering conference in 1996 in Danforth, Massachusetts, where one after the other speakers that we consider today to be the giants in the field of synthetic biology would go up on the podium and basically demonstrate how by reprogramming cellular systems, we can potentially produce whatever chemical we wanted. So we could potentially access chemistry we didn't even know that existed in nature. So that's basically what hooked me to this field. So the concept that we can take simple stuff, such as, for example, glucose, or waste like lignin, plastic, or carbon dioxide, and turn this stuff into complex chemicals, such as pharmaceuticals and nutraceuticals, or even chemicals that don't exist in nature, or even chemicals we don't even know that we can actually make. And on top of that, while doing all of this, we can also discover new mechanisms that exist in nature and develop new tools that can potentially be used, for example, in therapeutic applications and save lives. So all this basically blew my mind. And I mean, for me, what else was there to do besides synthetic biology, right? And this was perhaps where I made the mistake that I started talking about as a young investigator. So as an early professor in the field, I got very much convinced that the power of engineering biology lies with cellular engineering and lies basically with synthetic biology. And that everything else that comes after that was secondary at best. So during this past 20 years that I have been basically in academia as a professor, I've come to realize that yes, synthetic biology is indeed very powerful. And yes, we can program cells to do many things and produce many different chemicals. But unfortunately, so many of the things that we can actually do as synthetic biologists will remain at the lab scale and will never go any further than a simple shake flask, unless we have a more holistic vision of what is biochemical engineering. In other words, unless we will be able to talk to people after synthetic biology, for example, how we learn how to communicate and speak with people that do downstream processing, or people working on fermentation engineering, or people working on scale up, people who do techno-economic analysis, and people who do sustainability analysis. And to take this idea a little bit further, I will include in this list I just mentioned people that work, for example, in social sciences, and people who work in communication, because let's face it, creating a process, a synthetic biology process, is the very first step in a very complicated journey that will eventually get a product that came out from the synthetic biology project into the market and will have an effect to the general public. So to summarize what I basically tried to convey here, my words of wisdom for young people working in synthetic biology is seek out collaborators from various areas of biochemical engineering, but also beyond. Learn at least at a superficial level what they do and what their challenges are, and work together to make things happen in order to make basically synthetic biology the truly impactful engineering field that it is. So that's basically my introduction.
Oh my God. So wow. So amazing talk and inspiring insights. And again, you are so kind, I guess. Also, you cope with my 5-minute talk challenge excellently. And also, I'm very comfortable challenging all big guys, because even young people in conferences are challenged to give even 1-minute speed talk right before post session. So why not? Big names should deal with this challenge. I mean, giving 5-minute talk, and also do excel every time in this challenge. So thank you so much for your wonderful, brilliant, inspiring talk. Thank you so much, Mattheos.
Thank you.
Okay, so now the main speaker of today with a longer introduction. So Dr. Qing Sun joined the Department of Chemical Engineering as assistant professor in 2018. And in 2015, she obtained her PhD degree from the Chemical and Biomolecular Engineering Department at the University of Delaware under the mentorship of Professor Wilfred Chen. Obviously, I respect him a lot. He's one of the pioneers, of course. And then that means, she trained very, very well. So there's no surprise he, after that, joined one of the fantastic, labs at MIT, Professor Tim Nu's lab in Synthetic Biology Center at MIT. So she did postdoc training over there. And also she published tons of great papers in journal, including Nature Chemistry, Nature Communication, and ACS Nano, and so on. So I believe she will talk about this brilliant work, today. And her research interests are synthetic biology, of course, with advanced design and application toward microbiome redesign, mRNA therapeutics. That's important nowadays because the COVID vaccine, also biomaterial for environmental and biomedical application. Of course, this is the global problem we need to solve together. So if audience happen to be big name YouTube channel, or today big names potentially writing tenure letter, I believe you should write the strongest letter for her, because without doubt, and I believe she's the true rising star in synthetic biology. So Qing, take away. Thank you so much for your talk and time today.
Thank you, Tison, for the nice introduction. Yeah, I really appreciate the opportunity for young junior faculties to have the opportunity to talk to, I would say, public and our community, like synthetic biology community. That's really nice introduction, and thank you for the opportunity. Yeah, and thank you for Matthews to be my pioneer in the session. I saw your name. I was so excited, like we were connected, and you picked me as a young investigator to start talking about. And thank you for the great perspective. I will connect that in my talk. And also thank you all for the audience, including my PhD advisor, Wilfred Chen, who sit in the audience, and some of my lab members and other friends I know, including Xiaojun, Fang Chao, Kevin, and many others I cannot recognize the name right away, but thank you for sitting here. I will give my talk. I will try to keep it in the time frame by going into less details. Please let me know if you have any questions, and I will be happy to answer it throughout the talk. Okay, so now I'm ready. So the topic of presentation is synthetic biology for enhanced mRNA functions and animal performance. And this is synthetic biology young speaker series. So as we were talking about, so this is a picture that a lot of people like to use in synthetic biology. It's like if you have a toolbox that has DNA, RNA, protein, like bacteria or cells, or even like animals you have, what you can do with it. So what we can do as synthetic biology is actually, but after Matthews share his perspective about what real problem we can tackle with this toolbox. So our lab has a range of targeting programs and applications that from the range of macromolecules to bacterial strains to animals and plants. So today I'm going to focus on certain projects that we initiate that focus on the RNA, protein, and animal perspective, including bacteria. So in that case, the objective of today's presentation is a synthetic biology for enhanced mRNA functions and animal performance, with the first target of nucleotide engineering, where I'm going to talk about mRNA secondary structure and stability prediction in combination with thermostable mRNA therapeutics development. Then I'm going to switch the topic a little bit and talk about bacterial engineering and how we can use it to program animal physiology and behaviors. To start with, why we're interested in macromolecules, including DNA and RNA. So DNA and RNA are essential components of life. Understanding those macromolecules is critical to understand life and to re-engineer biological systems. Think about how much information is buried in there and how much engineering potentials we have with them. So one of the huge and obvious application is what we're doing now to fight against COVID-19. So COVID-19 is coronavirus disease 2019 that happened in 2019, that is still going on, that actually hindered my career a little bit and a lot more in my life. So one of the effective and efficient approach to fight against COVID-19 is actually mRNA vaccines. So how it works is we have those functional mRNAs that code for spike protein of COVID-19 virus, coronavirus, and encapsulate that in lipid nanoparticles for delivery. So Pfizer and Moderna, Biotech and Moderna are the two big companies that produced and deliver those vaccines worldwide to fight against COVID-19. It's very effective. However, one of the huge limitation associated with this system is mRNA thermal stability. That's why, like Pfizer has to be storing and distributing their vaccines at -80-degree freezer, while Moderna has to do it at -20-degree freezer, which is not a long resolution if we want to continuously fight against different viruses. And if we are going to be ready for the next wave of viruses that we don't know what they are yet. So those kind of situation somehow is closely associated with mRNA stability because it's easily to be attacked by like solutions, like iron in the solution or enzymes. During the approach, Dr. Das at Stanford University reported that there is a correlation between mRNA secondary structure and stability. So we all know like mRNA form this kind of secondary structure. So it turns out if they form this kind of loop structure that is left alone, it's more prone to be cutted. Well, if you form this secondary structure, it's relatively safer from degradation. It was this conclusion was also facilitated by Moderna experiment showing secondary structure actually help mRNA stability in mammalian cells towards final translation efficiency. With those knowledge, like we were thinking how we can restabilize RNA sequences by redesign RNA codon usage. So because we know that protein sequence can be coded by multiple DNA and RNA sequences, that means we can actually change the RNA sequence in there to optimize the stability while we don't change the translation capacity. So that the final protein coding results are the same. And so we hypothesize that the final immune response triggered by those vaccines, mRNA sequences should be the same. So the current challenge is that with the protein sequence, we can definitely reverse and change many of the RNA codings and test experimentally about how stable they are, what will be the efficiency of the translation, and what will be the immune response that will be triggered by the system. However, even if nowadays we have a lot of automation system including like automatic pipetting that can replace graduate students, it's still a lot of challenging because it's going to be low throughput. Think about how many variants we can produce using those RNA sequences, using the protein sequences, and also if we want to order every RNA sequence and test in the lab, the cost is super high. In that case, our lab are going to use deep learning to tackle this problem. Deep learning is a new class of data-driven modeling approach that has been utilized in many areas, including image recognition, natural language processing, and computational biology. Basically, how it works, it mimics how we learn from examples and find the pattern, hidden pattern that we cannot easily summarize. Its flexibility allows it to handle different forms of data with ease, and the neural networks are uniform function approximators that we can use a lot. So in that case, we're going to use deep learning, but what kind of technology we're going to use? So basically, we start with natural language processing. So this is a process where it codes a language encoding information. So it was used for translation, but nowadays we can use it for many other applications. Specifically, we're going to use transformer, which is part of the language processing architecture. So what is transformer? So it's a state of the natural language processing architecture, originally designed for translation so that it will capture all the information in the language, recognize and translate. So now it has been widely used in many other applications, including porting folding structures like AlphaFold 2. It can outperform human experts in highly complex video games. It also provides the art image recognition performance. So with that, we think DNA and RNA is actually a kind of language. That's why we want to try the transformer in DNA and RNA languages. So traditionally, there are many other like architecture neural networks that use for for DNA and RNA information processing, but the traditional way is using the recurrent neural networks that is really good at recognizing the local language, but lost is vision when it goes to long distances, because DNA and RNA can actually process a lot of information from long distance. So transformer compared with these recurrent neural networks is that it will capture the information regardless of the distance in the input or output sequences using attention mechanism. So in that case, we build the nucleic transformer neural network. How it works is that first we still use one-dimensional convolution that recognize the local information of the DNA and RNA sequences. And then we use the transformer to learn the global dependencies of the sequence. So in that case, our system can capture both the local information as well as the global dependencies. So first, with this network, what we can do? So we can use it to understand the RNA secondary structure as well as stabilities. How it started with was we were originally working with a transformer, but for DNA information storage, checking. And then we saw that Professor Das hold a competition on Kaggle. He actually provides a huge data set of how RNA molecules can be stable or not stable from sequence level. So it actually gave out 3,000 RNA molecules with stability information correlated. So we actually use our transformer to tackle this problem. So RNA is a little bit with DNA when we were only talking about information storage. RNA is single-stranded and highly promiscuous. So we actually use existing biophysical model so that we can get one layer of information and inject those information into our transformer model so that we can get thermostability information out. So that's how we are going to use a biophysical model. So we actually add into base pair probability as well as the structure and the loop information as an input to inject into the transformer system, as we mentioned in the previous slide. So we were able to get 7th place in this competition. I'm going to talk about our results soon. But we were, after post-competition, we used semi-supervised learning so that we were able to outperform the top solutions in open vaccine. So you can see that they have the public data and the private data. So this MCR-SE is a criteria that we can judge if the model prediction is good or not. The lower the better. You can see our semi-supervised model was able to further enhance the performance of the system. And we can predict reactivity at base pair with really good R-squared scores. So after we predict this, for our manuscript preparation, which is also available on arXiv, you can check it out. But we were trying to figure out what is the most important factor for RNA degradation, what is lead to RNA degradation and what make it more stable. In the end, we figure out that sequence and base pair probability metrics are by far the most important, like these two most important factors that help stabilizing RNA degradation. In that case, we can use these parameters to further design and the most stable mRNAs. So as mentioned, we are able to predict how stable the mRNAs are. Then like next from engineer perspective, as Mattheos has been talking about, we really need to figure out the problem and solve it. So after being able to predict how can we design stable mRNAs using our platform, so we actually have schemes here that we have the protein here that again is an example 5 amino acid sequence, how we can change it to reverse it to recode to mRNA sequence and optimize it by each species, by each codon, so that we can optimize it. But this is a huge computational task, because COVID, like spike protein itself is more than 3 kilo base pair. So that means it's a huge task for even for computational system. So we did a test using some original existing algorithm, including linear design. And we were able to, from the original GFP sequence, green fluorescence protein, we were able to figure out like one that is more stable. When you look at the structure, it's more stable. And our quick experiment shows that the GFP mRNA sequence is more stable compared with the original version. The half-life is increased by like twice under room temperature in these solutions. So because it's super difficult, so we are going to use reinforcement learning to design thermostable mRNA for vaccines. How it works is another algorithm where we originally we have the initial sequence, and then we have the reinforcement learning. It will first use biophysical model to predict secondary structure. Then it will feed back the base pair probability and all the parameters, the agent back into the transformer, and then we're going to predict again and feedback again, predict again and feedback again. So this is a quick example of 100 base pair example, and you can see after 16-step optimization, we were able to achieve majority of the RNA stabilized, but we assure that they encode for the same protein sequence as invariant parameters. So now what's going on in the lab, and what will the paper on the preparation now is that we're testing different protein sequences, including a 20 residual folding protein sequence and 1 L2Y for the simple structure. So to start with, we also use a COVID multi-epitope vaccine 141 base pair. It's still under testing, and it's much smaller than the COVID mRNA vaccine we're using now, and we're using green fluorescence protein as one test to see what kind of stability we can achieve, as well as how to if we can improve translation efficiency with the system. So previous data has shown that we should be able to achieve both higher stability and translation efficiency, but it's under test in our lab now. So I finished talking about the mRNA part. So this is where we actually started with when we were, before we were doing mRNA tests. We were trying to understand how DNA information is stored in the DNA sequence. So can we figure out the DNA promoter information from a, if you give me a random sequence, can we figure out if it's a promoter sequence or not? If we were receive some DNA information, can we classify if it's viral genome or not? So we actually use the E. coli promoter data site that is available online. We also use the viral genome data site that is available in the NGS sequences. So for time sake, I'm not going to talk about details, but again, you can find the details in our paper. So basically, just to quickly go through these 2 slides, we talk about how nuclear transformer can actually classify the promoter compared with what is current on the market. So that's our nuclear transformer compared with what other people are doing in this area. As well, we can accurately predict the viral genome classifications compared with other current algorithms. So the conclusion-wise for this part, I believe I'm good with time. So we are talking about how we can use deep learning combined experiment procedure to actually predict mRNA stability and to design more stable and highly efficient immunogenic mRNA vaccines. So in the end, we should be able to with our preliminary results, we should be able to produce this more stable and highly efficient system in the lab and see where we are going from there. And we were also able to classify the DNA information stored in DNA sequence, so that in the future, I hope, like with any DNA sequences, we were able to identify what's really the information stored. With that, after finishing up the first objective about nucleotide sequence engineering, talking about DNA sequence identification, mRNA secondary structure and stability prediction, as well as thermally stable mRNA therapeutics development. I'm going to switch the topic a little bit because, as I mentioned, we are working on different levels of challenges to see how we can engineer bacteria to program animal physiology and behaviors. So why program animal physiology behaviors? So synthetic biology has been tackling different problems, as mentioned, from DNA, like protein level as macromolecules. We're also talking about how we can re-engineer bacteria or mammalian cells from single cell level with genetic circuit or different programming, like metabolic engineering. We also talk about bacteria community or like mammalian community, where how they are programmed, how they can stabilize and work as what we wanted to do. And we also talk about plants, like in synthetic biology area. However, one of the challenges that has not been done much is how to program animal physiology and behaviors. So there are many applications that could fit in this area, including pest control, agricultural applications, and like healthcare applications. However, programming animal is always challenging. So think about all the gene editing cascades like we need to build and we need to deliver for this kind of animal physiology and behavior programming. And if we need to go to embryo engineering, that may also cause some ethics problem, including how we're going to handle these genetically modified animals. So those are all the challenges associated with animal programming. So we were like, we want to do this, look at animals, but don't want to directly engineer those animals. Can we actually, from another approach, can we engineer those associate microbiome and program animals? So we all know that bacteria and animals have coexisted for millions of years. That's where bacteria, or we call it microbiome, can reverse the animal nutrition, immunology, behavior, and many other things. I think this picture shows the best about what we can potentially do with microbiome and animals. So in that case, so our goal, we are going to do baby step. We don't want to program the most complicated animals in the first step. So we're like, if we want to program a simple animal, for example, if we want to program the physiology like lipid metabolism and behaviors, so can we actually start with this bacteria where it can sense and compute the signal and output the actuator which talk to the animal so that we can program animal in the end. So in the end, we don't program animals, but we use engineered bacteria inside. So to achieve this, we definitely need three things as demonstration. Number one, we need genetic circuit in animals. Number two, we need communication tools between the bacteria and animal. And in the end, we need an animal model to show the how our system works. So synthetic logic gates in bacteria. So I use one of the figure from my postdoc advisor, Tim Lu's lab, showing like how nowadays in bacteria, we can already program bacteria with multiple synthetic logic gates, including like and or nor, not A, not B, A imply B, B imply A. I could show many other figures, including Dr. Chris Voigt's lab showing how 3 layers of different like genetic circuit, or from Dr. Moon lab showing different Q-switch constructions. So these are all synthetic logic gates that can already well started in bacteria. And for the animal model, we use C. elegans. It's like relative similar structure. It's 1 millimeter round, like transparent worm that has a lifespan of 2 weeks to 1 month. Although it's simple, but it has complex functions. So like it has different lifespan, the life stage where we can study the development, sexual behaviors, behavior, longevity. So this is a C. elegans model. And in terms of DNA, in terms of bacteria-animal interactions, one of the, we were looking at what kind of communication between them. So first, what is quite unique for C. elegans is the RNA. So RNA interference happens in C. elegans, and it can be delivered by bacteria. So what happens is bacteria can produce this single-stranded or double-stranded RNA, and then C. elegans, by feeding on those bacteria, can grind up the bacteria, pick up the RNA, and trigger RNA interference in C. elegans that will mute certain genes in C. elegans. So that's quite unique for C. elegans, but super efficient. Another intermediate we talk about could be metabolites. That also happens in human and many other animals where bacteria can produce these metabolites, including short-chain fatty acid or neurotransmitters as examples that can modulate C. elegans physiology and behaviors, including like fat storage or behavior control. So both of them can be done by feeding those bacteria to C. elegans or colonize them in C. elegans gut. So with those structures, now we have a clear goal of having those bacteria with logic gates, where they will take up the input signals. We use two arabinose and doxycycline, which are the two well-known like synthetic circuit input system. Bacteria take up those signals processed with logic gate, and then give RNA interference phenotype to C. elegans. So we use green fluorescence RNA interference. For example, we use C. elegans with green fluorescence, and if the RNA interference works well from bacteria to C. elegans, we should be able to see the GFP silencing C. elegans as a quick demonstration about like bacteria can actually trigger RNA interference in there. So again, like we use C. elegans with GFP, which is artificial, but in the future we will move to well-tapped C. elegans where we don't need to see any gene modification on the animals already. So the first step, as planned, we're going to have first on and off system of GFP RNA interference. So how that works is we will have double-stranded GFP RNA that is triggered, that is transcribed by T7 promoter from both sides of the DNA, so that we will have double-stranded GFP. That is how bacteria is designed, it's engineered. So the first step, we will have a plate that with IPTG or no IPTG, and then we will put the bacteria on there to enable the transcription of the RNA. And then we put on the worm. So the worm will feed on C. elegans and see what will be next step. We hope to see the silencing of the GFP in there. So I had a lot more data, but for time's sake, we did a lot of work here. My first student, Bajan, is here, who struggled a lot with it. We'll see a lot of the different phenotype with RNA interference. But in the end, we were able to screen because at that time, the on and off status of this genetic circuit was super difficult. Either it was all turned off or either it was on all the time. So we screened a bunch of double-stranded GFP and single-stranded GFP with different lengths. We were able to figure out a certain length of the single-stranded GFP RNA were able to trigger our GFP RNA interference in C. elegans. We decided to move forward with single-stranded 200. I feel bad because my students spent like almost 1 year figuring this out, but I have more exciting results to show in the next several slides. So after we figure out single-stranded 200 base pair GFP is the best target, best like RNA we can produce to trigger RNA interference in C. elegans, we move forward and construct the logic gate. So I believe the majority of the audience are from synthetic biology, so you should be familiar with logic gate, but I should talk about it, yes. So we have two different promoters, like tight promoter and bad promoter, that control the T7 promoter C-terminal and N-terminal like production. Only in presence of both signal arabinose and ATC you should be able to see T7 polymerase, which will trigger our GFP RNA production. So the hypothesis of this AND logic gate is that only in presence of both arachidons and ATC signals, we should be able to see GFP. And only in this case, we can see GFP silencing in C. elegans. After a lot of construction and testing, we use a similar strategy where we have the plate, we put different signals in there, including 1 signal, 2 signal, or no signal. And we put bacteria in there and we put C. elegans in there. In the end, we were able to see that only in presence of both signal arachidons and ATC, we see like the silencing of GFP proteins in fluorescence worms. So you can clearly see the difference. And we did a quantification that shows that we were able to silence the GFP signal from C. elegans using these logic gates and the logic gate. And after AND logic gate, we move forward and did the OR logic gate. So the OR logic gate is that we put the T7 RNA polymerase under control of both TAC and BAD promoter. In this case, in presence of any signals, we should be able to trigger T7 RNA polymerase production and in the end produce a single-stranded GFP RNA, and that will trigger the GFP silencing in the C. elegans worm. And we can see here is exactly as what we expected. So in presence of any signal, including arachidons or ATC, we were able to see the silencing of the GFP from the worm. And in presence of both signal, again, we see that GFP signal is gone from the fluorescence worm. The data also supported that we were able to achieve OR-gate programming on the GFP expression profile in C. elegans. So every time I talk about GFP, I would say it's a cute and little try on certain phenotype before we move to real behavior and phenotype, because GFP is just one gene there, does not have any real functions. So what we next do, we want to control certain behaviors in those, and so we picked UNC22, which is a gene that regulates muscle contraction. So if we silence UNC22, it will lead to a twitching behavior in C. elegans. So again, this is the Nature Communication papers we published in 2021, showing how this behavior programming works. Just a quick show that if you have normal worms, we already fixed them, so they don't take much. But if you have those UNC22 silence, they started twitching because its muscle contraction is out of control. So that after those, we can also see we were able to program those twitching behavior using AND gate, showing that only in presence of both signals, we can see the twitching behavior in the worm. It's actually 100%. We were super surprised by how well it worked. And we were also able to achieve the twitching phenotype with OR-gate. You can see in presence of any signal or both signal, we show the twitching worms in our experiment. And after those twitching behavior, we got more excited. We were like, yeah, we can also, one of the hot topic for me is how to control the fat distribution in your body. And we use SBP1, which is a homolog for two steroid regulatory alimentary binding proteins. It regulates lipogenic genes. So it silences, if we silence SBP1, it will lead to reduced fat storage. You can see original, it distributes a lot of fat along C. elegans body. And if we trigger the RNA interference, actually they lose a lot of fat from their body. So we use a little bit different dye and filter, so the fat is stained in a different color, but this is an active control. That's how normal worms store their fat. And after we put this AND gate SBP1 bacteria with C. elegans, we can see in presence of both signals, we were able to reduce the fat storage in C. elegans as AND gate output. And our quantification support our data, showing that we were able to achieve this AND gate programming using RNA interference and using AND gate in bacteria without changing anything on C. elegans. And we were also able to achieve OR-gate programming of fast storage in C. elegans, you can see in presence of any signal that fits bacteria that will trigger C. elegans fast storage change. And our data again showed how well the OR-gate is working. So the conclusion for this session is that we were able to program animal physiology and behavior through engineered bacteria. This signal is ready for the bacteria so that they can pick up the signal and program the production of the RNA. And also we feed those bacteria to C. elegans, they were able to pick up those signal from bacteria where they respond to the signal. We talk about GIP silencing, behavior control, that is twitching behavior, as well as metabolism control. So again, it was published. It took my first student several years to finish this project. So I mean, it looks short on the presentation. I believe that's the thing for all the projects. So after the RNA interference, we really started thinking about the question, RNA interference is specific for C. elegans, right? So if we use those bacteria in human, definitely that's not going to work. So next step, we're thinking how we can make it a more universal platform. So as we mentioned, metabolites are one of the key components that bacteria use to communicate with other animals, including human beings. So our plan is to identify intermediate metabolites between bacteria and animal for the animal longevity, development, behavior, and more. And after identifying those metabolites, we can certainly use genetic circuit and metabolic engineering to program bacteria intermediate production. In that case, we can achieve higher level animal programming using our platform. So question we're asking is that we found that bacteria can be like beneficial or harmful. So we found that like bacteria, for example, we always think probiotics consumption is good, and we always think like different bacteria, if we have the good ones, it will also always help development and longevity. But what we found in our lab is that some of the bacteria can facilitate the beneficial effect of probiotics by making C. elegans live longer. And some other bacteria make probiotics become harmful by, like those worms after consuming the probiotics, they actually have a shorter lifespan. So this is the phenotype we found. And we also found that different bacteria change C. elegans response to temperatures. So we are super excited to, like with this phenotype, we want to figure out what is in those bacteria that triggers like animal in using C. elegans as a model animal to see how they respond to probiotic consumption or how their development, longevity, and temperature change has changed when we have different bacteria to start with. C. elegans is a really good model because originally it feeds on bacteria. That means it uses bacteria as macronutrients as well as micronutrients, which we talk about as metabolites between bacteria and human beings. So the question we're asking is, what are the genes from bacteria that lead to this I would say physiology change, and what change they have made on the animal that like genetic level to for this phenotype different, and are they universally applicable to all animals, including human beings? With those questions, our lab started like a screening platform where we have E. coli with single gene not out mutant library KO collection. I believe many of you are familiar. So it's like single species, but each species has one gene mutant. Like they can survive, but has one gene mutant. So we have almost 4,000 genes in the lab as microbiome representative, and we have C. elegans as host representative. And we feed those microbiome to C. elegans and see how they respond to the probiotics differently, including longevity and development. That is one of the targets. So the motivation again is that we found probiotics effectiveness varies from person to person based on microbiome composition. So we want to figure out what microbiome components in there that trigger those different like effectiveness. So other than probiotics effectiveness, we're also interested in how this microbiome affects the C. elegans or animal response to temperatures. I will quickly show some preliminary results. It's really prelim, showing that after we did some screening on E. coli probiotics, we use lactobacillus and C. elegans, we were able to identify certain genes using this screening approach, using kill collection, like 4,000 genes, we put that on the plate and feed them with probiotics and check on the development and the longevity. And we repeated screening to figure out, and we were able to figure out that for this, we identified 29 genes in E. coli that help C. elegans survival on probiotics. We are still studying like what genes help C. elegans the longevity on probiotics using the same screening platform. So this is a really new area for us because we're really looking at metabolism and both on bacteria and C. elegans level. We found the vitamin B, iron balance, cysteine biosynthesis. So we are really working hard to figure out the mechanism level on what's going to happen next. So the conclusion, I jump into a conclusion of the overall slide because I think it's almost 40 minutes. I'm right on time. So our lab is working on hope that reply to Matthews' thoughts on application level, like we're trying to see what mRNA therapeutics development can be better using our synthetic biology tools, including deep learning and experiments, how we can re-engineer bacteria to study bacterial-animal interaction and to program animal physiology and behaviors in the future. We have other projects I did not include, but that's actively going on, including how to degrade plastics using our engineered bacteria. I believe many of the faculty in this session are working on this plastic degradation. I'm happy to talk about if you have any thoughts. So we got the NSF every to work on this. And we're also working on some enzymatic DNA synthesis platform for a more efficient and accurate DNA synthesis platform. So in the end, it's acknowledge slides, which I always say from my heart that I really appreciate the help of the funding resources, like they trust me to push for the project, including NIH-R1 from NIAID to develop thermally stable mRNA vaccine that I got it last December. I was super excited. We got the Texas A&M President Excellent Fund. It's also a huge, like 1.8 million for 8 faculties to develop, like to study second structure and stability relationship. We also get NSF every to degrade plastics using microbial communities, using computation tools and engineering tools. And more importantly, I cannot do anything with my lab. So I'm on my 4th year on my tenure track. I have 8 members. It's a not easy process. I struggled, and my students, I really appreciate that they joined my lab when I'm still a junior faculty. So they did a lot of work with me. We, after the pandemics, we get out to eat a lot, and I hope to recover that next week. We're going to have a group meeting and group eating together. So Bai Zhen is my first student who did the majority of the microbiome work. Shi Jun did the majority of the deep learning work, and Tracy is my technician and lab manager who is working on the mRNA stability validation experiment part. With that, I really appreciate your time sitting here listening to me for 40 minutes. I'd like to take any questions that you may have and any feedback for my career and the lab and vision and any connections to the industry or academic world. Thank you all.
Amazing. Amazing, fantastic talk. Wow. So I love, of course, Thursdays the most. I mean, since this seminar started. Also now you will be loved by many people, including my teenage daughter and my wife, because you now can control worms in your
Thank you.